AP 120, Chapter 8, Topics, Muscle Structure and the Sarcomere. So, here is a muscle organ, here's a bone. So what does the tendon here do? I hope you said attached muscle to bones. So most tendons in the body are attaching muscles to bones. So here is a look at a muscle organ that has been cut, as it were, to see internal structures. So if we look into the section here of the skeletal muscle, we see that there's all these little packets of things bundled up in the muscle. These packets are referred to as fascicles. So there are many, many fascicles bundled up in a muscle. And if we look inside of the fascicles, we see that there are smaller structures found bundled up in the fascicle. And those smaller structures are the muscle fibers, also known as muscle cells. So muscle cells are bundled up into fascicles. Fascicles are bundled up into skeletal muscle organs. And these bundles are partially formed by sheets of connective tissue. So wrapped around every muscle fiber is a sheet of connective tissue called endomysium. So endomysium, shown sort of as a white little circle here, is the connective tissue surrounding individual muscle cells. And surrounding the fascicles is a connective tissue structure called the perimysium. So perimysium, shown as a white line here, surrounds and forms the fascicles. And then finally, there is a sheet of connective tissue that wraps around the entire muscle organ, and that is called the epimysium. So epimysium covers the muscle organ, perimysiums cover the fascicles, and endomysiums cover the muscle fibers. Here we are taking a closer look of a muscle cell, and we see that muscle cells are filled with these rod-like protein structures called myofilaments. So you've got many, many myofilaments bundled up inside of skeletal muscle cells. And these myofibrils are made of protein filaments, also bundled together. So that means a muscle organ is bundles of bundles of bundles of bundles. You got myofilaments, the protein filaments bundled up to form myofibrils, myofibrils bundled up inside the muscle fiber, muscle fibers bundled up to form fascicles, fascicles bundled up to make skeletal muscles. All right, here is another close-up view of a skeletal muscle and a skeletal muscle cell. And so the cell, to start with, some vocabulary change. The cell membrane of a muscle cell is known as a sarcolemma. Instead of cell membrane, we say sarcolemma. And then the cytoplasm in a muscle cell is now called sarcoplasm. So sarcoplasm is the cytoplasm within the muscle cell. Sarcolemma is the new name for the muscle's cell membrane. There are also some structures specific to muscle cells. So for instance, we have these tubes made up of cell membrane, sarcolemma, that go into and pass through the muscle cell. So transverse tubules are tunnels, as it were, going through the width of the muscle fiber. And so the fluid inside the transverse tubules is the same extracellular fluid as surrounding the rest of the muscle cell. And also within the muscle cell is this membranous structure called the sarcoplasmic reticulum. And the sarcoplasmic reticulum, its main function is to store calcium ions. Here is a closer look. We see that the transverse tubules have the terminal cisternae, the terminal ends of two different sarcoplasmic reticuli lying adjacent to it, so that you have these three structure that reoccurs again and again. And so the structure of three is called a triad. So a triad is two ends of sarcoplasmic reticuli abutting one transverse tubule. All right, here is the myofibrils, again, bundles of protein filaments known as myofilaments. turns out the myofilaments are made of two kinds of protein filaments. It's made of thick filaments and thin filaments. And it turns out the thick filaments are thicker than the thin filaments. Now, the thick filaments are made of one type of protein, myosin proteins. So hundreds of thousands of myosin proteins make up a thick filament. And then the thin filaments is made of three different proteins. Made up of actin, which is represented by these spheres. It's probably the most common of the proteins in the thin filament. Then you have tropomyosin, represented by these green threads, and then troponin, represented by this hungry blue snowman. And these filaments get arranged in a structural unit called a sarcomere. So here is a sarcomere. We see there's the thick filament in the center. We have the thin filaments on the sides. The thin filaments are attached to a structure called the Z-disc. So the Z-discs form the ends of a sarcomere and also attaches to the next neighboring sarcomere. Another structure is the M-line. The M-line in the middle is what the thick filaments are attached to. As you can see, the myofibril is made up of sarcomere after sarcomere after sarcomere after sarcomere. So you have hundreds of sarcomeres stacked, connected to each other, forming the myofibril. And what's pretty amazing is when you look at the muscle fiber under a microscope, you can actually see these patterns of dark and light. It is the striations that skeletal muscle fibers possess. Here's a super close-up view of the electron microscope of the striations. So here is the Z-discs. Down the middle here is the M-line. You got the thick filaments in the middle, thin filaments at the ends. So anatomists love to name everything, and so different regions of the sarcomere have their own distinct names. So for instance, the A band is the width of the thick filament. So A band is the complete width of the thick filament. Then the I band is where you only see thin filaments. So there's no thick filaments, only thin filaments in the I band. And then the H zone here in the center is where you only have a thick filament. So A band, length of thick filament, I bands where there's only thin filament, and H zone where there's only thick filament. And these regions are important because they help to show how the sliding filament theory works and how movement causing the sarcomeres to get shorter is what leads to the muscle contraction. So here we have the biceps brachii muscle at rest, all relaxed, and you have the sarcomeres as wide as they can be. You got lovely H zones, I bands, and of course the A bands. Then when you start to contract the muscle, what happens is the thick filament pulls the thin filaments toward the M line. And of course, when the thin filaments get pulled toward the M line, that drags the Z discs so that the overall size of the sarcomere gets shorter. And this means there's now more overlap. So the H zones get smaller and the I bands get smaller. And that is with a partially contracted muscle. And then when the muscle is fully contracted, the thick filament has pulled the thin filaments as far toward the M line as it can. There's now complete overlap. So there's no I band, no H zone, only the A band. So the smallest the sarcomere gets is the width of the thick filament. So fully contracted, we get the smallest sarcomere. Start to relax, the thin filaments slide back, separating. Now we got H zones and I bands again, get fully relaxed. The I bands are now back as far as they can get, and we have the sarcomere as wide as it's going to get, the muscle as relaxed as it's going to get. And again, this is sliding filament theory because the thin filament is sliding toward the M line across the thick filament, so it's sliding. The thin filament, the thick filament, they stay in the same size. Their sizes do not change. Only the size of the sarcomere changes, and that's by the I band and H zone getting smaller as the thin filaments slide toward the M line. And that is it for this part of the lecture.